There are two different broad classes of stem cells. Embryonic stem cells, isolated from an 8 to 10 days old embryo, and then adult stem cells. So embryonic stem cells and adult stem cells, which are stem cells after birth.
Christian Drapeau. He is a stem cell scientist, an author, the creator of the first stem cell supplement. He holds a graduate degree in neurophysiology and has been involved in medical research for 30 plus years, the last 20 specifically dedicated to stem cell research. The author of 5 books, including the bestseller Cracking the Stem Cell Code. He has also published dozens of scientific papers, and he's coined the phrase endogenous stem cell mobilization. He is a scientific advisor on many companies, and he is currently the CEO and founder of Collagen, where he developed the most potent stem cell mobilizer, STEMREGEN.
Any kind of health problems, something that is broken needs to repair. Your repair system are your stem cells. So at the core of absolutely anything that you want to do to improve your health, stem cells are the basics. Probably my first one following this would be...
Hi, my name is Chris Burris. I'm a longevity expert and the chief scientist at a lab focused on a longevity molecule. My primary focus is to help people live longer and better lives with science. Make sure to subscribe and share so you and your friends can live longer and better, so you and your friends can live beyond the norms.
Hi, and welcome to another talk on the Uncovering the Secrets of Longevity, Live Beyond the Norms Health Summit. We're talking about stem cell stimulation and longevity. So stem cells are extremely popular right now. So if you do a Google search on stem cells, you got 394 million results from Google. And the thought of herbs, extracts, even algae that can increase stem cells in your body sounds fantastic. And if you think about all the things stem cells are believed to be important for in our bodies, it sounds like maybe even unlikely. But that is why we're here today with Christian Drapeau. He is a stem cell scientist, an author, the creator of the first stem cell supplement. He holds a graduate degree in neurophysiology and has been involved in medical research for 30 plus years, the last 20 specifically dedicated to stem cell research. The author of 5 books, including the bestseller Cracking the Stem Cell Code. He has also published dozens of scientific papers, which I've referenced in some of the questions I put together here. And he's coined the phrase endogenous stem cell mobilization. He is a scientific advisor on many companies, and he is currently the CEO and founder of Collagen, where he developed the most potent stem cell mobilizer, STEMREGEN. Christian, welcome to your talk.
Thank you, Chris. Pleasure to be here.
So before we kind of jump into a lot of stuff, I want to understand your backstory. I think is fascinating. How did you come to study stem cells?
Well, as you just mentioned, my background is brain research, neurophysiology. I was doing research at the Montreal Neurological Institute on memory and epilepsy. And I was hired by a company at the time in 1995, a company selling a dietary supplement that is fairly well known, I believe, blue-green algae from Klamath Lake in Southern Oregon. And DSHEA had just been passed, the Dietary Supplement Health and Education Act, that basically says if you want to sell a product, you need a natural product, a dietary supplement, you need to have science to back up any of the claims that you're making. So I was hired to document the health benefits of that plant and the mechanisms of action. And as I'm studying this plant, we quickly identified many mechanisms of actions, benefits, but I came across people who reported pretty astonishing benefits, but from a scientific standpoint, the most challenging or the most intriguing for me was some reported great benefits touching the pancreas, others the heart, the lung, the liver, the brain, joints, so many different aspects of human health that the question was, what is this plant doing to touch so many aspects of human health? And one thing's leading to another without a good explanation. One day I came across a paper documenting how stem cells had been observed going from the bone marrow to the brain and becoming a brain cell. You go back in 2001, stem cells are only precursors to blood cells. And if anything repairs in your body, it's not your brain. And so that study was to me like very, very groundbreaking. So I went in the literature, looked for anything else I could find. I found an article documenting stem cells being able to go to the liver, becoming liver cells, same thing in the heart. So I thought, if stem cells can become brain, liver, and heart cells, Why not pancreas, lung, skin, and the rest? Makes no sense that they can become just those three and not the rest. So it's just a matter of time for science to discover that they can become everything. And if they can, they are the repair system. So we published an article in 2001 in a journal called Medical Hypotheses, suggesting stem cells are the natural repair system of the body. And in the back of my mind, the thought was, what if that plant is working as a stem cell mobilizer, releasing stem cells from the bone marrow? So we acquired the instrument to count stem cells. And to make the story short, although it was a long answer, this plant, blue-green algae from Klamath Lake, actually that's what it does. It acts as a stem cell mobilizer. And it took my entire career in the direction of stem cell research.
It's amazing how scientific kind of discoveries tend to be these serendipitous of like, well, I've got all these results and I just can't really explain them. Oh, stem cells valuable. Maybe I can put these together. I heard you in one interview that original publication, I guess 2001, wasn't that well received by the scientific community?
Well, it's very yes and no. We need to understand the science that I used to propose this hypothesis. I didn't take that out of thin air. There were already many studies already done at that time, clearly indicating that if you really put all these dots together, it's kind of telling you your stem cells are your repair system. But in a world where stem cells are only known to be blood precursors, to put forth an idea like this, there are a lot of people that are going to say this is nonsense because this is not what I learned in college. Guess what? This is not what I learned in college either. And that's what scientific development is. You know, you just learn stuff and then you later discover something new. So the idea that it was not well received is that it took quite a bit of time for the idea, first after being suggested, to kind of percolate through the scientific community and through the world. It's clear to my mind, I could not have been the only person in those days thinking that stem cells were the repair system. But I did not have an academic position, a reputation to risk. I did not have any, I could just freely suggest what I thought was there without any fear of any repercussions. So that's probably why I was the first to publish on it. And I remember maybe 7 years after, so we're around 2007, and I'm giving a presentation on that data. And there's a doctor standing in the room and says, all of this is nonsense. Let me tell you something, son, because I think nobody has told you, and you just need to hear it. You're just an idiot. And it was funny because the data was so solid. There was so much data. It was absolutely undeniable. We were just hitting, that's what dogmas are, the things that you learn in college. And if you hear something different from somebody who's not a university professor, then it's not true because it doesn't fit with what I learned. That was the period of transition that was kind of almost funny to go through. Sometimes I would say annoying, but yeah.
Yeah. So you found this one component, right? A blue-green algae that stimulates stem cells. And maybe we can talk about that a little bit more. But I know that you found others. What was your process for identifying others and why'd you even go down that path?
Well, the first one was this blue-green algae. Now, when you tell the scientific community, I have a plant that is putting more stem cells in circulation. First answer, well, if you don't have a mechanism of action and an active compound, this may just be a fluke, may just be like a weird observation. It may mean absolutely nothing. Let's say it means something. You put more stem cells in circulation. Let's say that that's a fact. Well, what does it mean for your health? Maybe it means absolutely nothing. And then the other one is maybe it's actually bad for you. So it took about 5 years of research to establish that putting more stem cells in circulation, it's real. We have the mechanism of action. We have the active compound, and we have the proof of concept that it actually helps repair. Once all of this is done, during all that time in the back of my mind, if really stem cells are the repair system and we evolved in symbiosis with the environment, there cannot be just one plant having an effect on stem cells. Just like there's not one plant having an effect on our immune system. Mm-hmm. So how do you find them? You simply look at whatever has been documented over the years, over the centuries, what has been known to be good for many kinds of health problems. It's your typical adaptogens. Plants that do many things for you, but we don't know how they work. So think of medicinal mushroom, goji berry, ficoidan, those kinds of plants. So we went and studied those. And we found that they all, at the end of the day, they all have an effect on stem cells. Then I went to remote areas of the world. Let's say we go to Madagascar, Papua New Guinea, South America, in the Amazonian rainforest, on the Tibetan plateau. What are they using there? They don't have access to plants, that are like medicinal mushroom. You don't have access to them in South America or in Madagascar. What are they using there locally? And that's where we found probably some of the most potent plant that stimulates stem cell release from the bone marrow. So it was this kind of quest that led us to study other plants and find other interesting plants.
That's very cool. So one of the questions that came up for me was this initial blue-green algae that it just found somewhere that it doesn't stimulate pluripotential stem cells. One, what are pluripotential stem cells? And maybe talk about the different kinds of stem cells, but also, is it a missed opportunity for this blue-green algae not to stimulate those?
It does stimulate pluripotent stem cells. We need indeed to talk a little bit about the different kinds of stem cells. Probably what my statement was here that suggested this idea that they did not stimulate pluripotent stem cells is that it's, at the end of the day, of the plants that we have documented, it ends up being probably one of the least effective. So we have more potent plants now that stimulate stem cell release from the bone marrow. But of those that are working, on AFA, so that plant is documented to stimulate the release of stem cells from the bone marrow. These stem cells are pluripotent. Some people may say multipotent. Quite frankly, I think that they are much more versatile and potent than at times we think. Science likes to put label on things, and I make an analogy once that maybe is too simplified as an analogy, but somewhere I think it gives the picture. It's like taking a child who cannot, an infant, you call him Bob, and at 4 years old, Bob is starting to walk. Well, wait a minute. Bob was not walking. Now Bob is walking. So let's call him like Jim. And now 10 years later, he's running, he's playing football. Wait a minute. At 4, Bob was not running around. Or Jim was not running around. Let's give it another name. I think we look at the same thing at different phases of their lives as they transform very rapidly. And we think that they're all different things. I think that there's way more, oneness, if you want, in all these different types of stem cells that we document scientifically as being different stem cells. This being said, there are a few different stem cells. Those qualified as pluripotent, like, for example, V cells, very small embryonic-like stem cells, are stimulated by this blue-green algae, both in their number in the bloodstream and also their activation. So it's a short answer for something that can be fairly complex, but they do stimulate pluripotent stem cells and provide that additional heating power, if you want, to the body.
And so there are, is it 3 or 4 kind of classes of stem cells that are currently kind of in the literature? And I appreciate what you're saying is like, they're probably more similar to each other and maybe just different phases of the same thing, but is it 3 or 4? Am I right in that?
It depends how you want to look at them and how deep you want to go. So I would say at the onset, there are 2 different broad classes of stem cells. Embryonic stem cells, isolated from an 8 to 10 days old embryo. And then past that stage, it goes to fetal stem cells. There's no fetal stem cells available. It's not a thing at this point. And then adult stem cells. So embryonic stem cells and adult stem cells, which are stem cells after birth. So in our bodies, all we have is adult stem cells. In this class of adult stem cells, you have hematopoietic stem cells that make blood cells, and then mesenchymal stem cells that do pretty much the rest in the body. So in those so-called mesenchymal stem cells, but again, the difference between the two sometimes is not as clear as we would like it to be. You have substem cells. So you have your endothelial progenitor cells, for example, that can become blood vessels. And so adipose stem cells are also mesenchymal stem cells. Umbilical cord stem cells, which are not from you, but they also are adult stem cells. So these are other types of stem cells that are available in the marketplace. The last one that you can see when you do a quest or a search on Google is those so-called V cells. V cells were identified early on, let's say 2001, 2002, but they're very small. So when you take your blood sample, spin your blood, they're so small, they move with the platelets. They're the size of a platelet. So they normally are collected with the platelets. So when you do a platelet-rich plasma, platelet-rich fraction, they contain those small stem cells. And how you identify that they're stem cells? Well, they have DNA and they have the marker for stem cells. And these small stem cells are actually the most potent in the human body. So these are sort of the general stem cells that we find in the human body.
That's interesting. So they're in the PRP preparations that you then turn around and inject into, parts of your body. Typically, I actually had some done recently for a tennis elbow and a knee pain that I've had for some time. So it's actually is getting some level, the smallest size of the stem cells into that PRP.
Correct. They will historically, technically tell you, no. This is not stem cells. This is platelet-rich plasma or fraction, and it contains only platelets and growth factors and different things, that are there in the plasma. But it's not correct. They actually do have stem cells, and the repair power most likely comes, let's say 75% comes from those stem cells, and then accompanying growth factors that are already found or released by these stem cells.
Interesting. So earlier you said that you've got proof that these stem cells help repair. And I think I heard one of your talks where you're describing it. I thought it was fascinating. What is that proof?
You started by saying, you do stem cells and you said several 100,000 hits coming out. Go to PubMed, the library of the National Institutes of Health, and you will have, 500,000 articles showing up. It's unbelievable how prolific this field of research is. And a lot of these research document our stem cells repair. So one study that we did at the time, it was with the blue-green algae, is to inject cardiotoxin in the muscle of a mouse, in the leg muscle. So it kills muscle. It's a mouse model of muscle injury. And then we basically gave these mice blue-green algae, but before that, we injected them with stem cells that were tagged with green fluorescent protein. So if any one of these stem cells become a muscle cell in the healing process, that muscle cell will be green when you put a light on it. So we let the animal recover. 6 weeks later, the muscle is back, completely reformed. The leg has mobility, but you just shed a light on the muscle and the entire muscle is green, telling you that this entire muscle that has repaired right now came from stem cells. So really what happens is that since the day we're born, the question is, what's your proof that stem cells repair? And honestly, the real answer is that it's always been repairing since the day you're born. You're alive today because the stem cells do that job. It was just not visible until the time when we had markers to follow this phenomenon in the blood. So I can go through hundreds of these studies, but essentially stem cells are the repair system. If you have an injury, that injury will trigger the release of stem cells from the bone marrow. Then the injury will release other compounds that will attract stem cells to that area, will trigger their migration out of the blood into that tissue. And upon cellular debris, contact with cellular debris of that tissue, stem cells will multiply and transform into cells of that tissue. It is your innate repair system.
Yeah, I think it's such a great, sometimes when you get into these biological situations, to get a good visual can be challenging, right? So you've got these fluorescent stem cells, right? And now you have this fluorescent muscle. It's not rocket science to figure out what happened in this particular situation. That's pretty amazing. I also saw a video that you did where you talk about taking a heart and stripping away the muscle of the heart. Talk a little bit about that. We don't have the visual, but just to describe that I think will be powerful.
Yeah, it's an amazing study, and I'm using this just to really shock the minds, because we talk about stem cells, we accept that they're regeneratives. It's all great, but I don't think that it really sinks in people's mind the degree to which these stem cells are regenerative. And that study really illustrates that. So it's a group of scientists, a group of Dr. Doris Taylor, fabulous scientist in that space. And she took the heart of a mouse, digested that heart with proteolytic enzymes that digest protein, muscles. And after 12 hours, all the muscle is completely gone, digested. The only thing you're left with is the soft skeleton of the heart, like the connective tissue that is holding the heart.
And in that video, it just looks like almost a ghost of the heart, right? Like it's
Correct.
just the clear material that's left over.
Correct. Look, think of a glove without a hand in it. It's just hanging. And then she took stem cells from that animal and just like laid these stem cells on that soft skeleton that still has the biological trace that it was the heart. So the stem cells recognize that, and then they start to transform in the heart. And what is beautiful is that not only do they transform in cells of the heart, but they transform with the entire architecture of the heart, so that at the end of a few weeks, you have a fully formed beating heart in a test tube. And that is the power of our stem cells. We have those stem cells in our body. That's what they can do.
Remove all muscle from a heart framework, add stem cells. Those stem cells recognize, because the framework still has the encoding of, hey, you're in the heart area and you need to be here, or however this magic works. And then they turn into the muscle cells, and then collectively they do what muscle heart cells do, which is begin beating like contracting synchronicity. I mean, I love that demonstration. I thought that was amazing. Let's shift gears a little bit. And let's talk about the role of stem cells in disease formation.
This is something that, to me, it's probably the most relevant of all that that research on stem cells, and it came to a large extent sort of incidentally. We study repair, and we suddenly discovered that stem cells do something else in addition to repair, because most of our lives we don't have something to repair. We're just aging and moving along, and we're fine. Of course, we experience aging, but we don't have anything broken. But stem cells play an essential role even when nothing is broken, and that was seen. Let's take the example we just talked about before, where we trigger an injury to the muscle, and then we release stem cells that are green. So you see the entire muscle being reconstituted, and now the muscle is green. But if you keep some of these animals alive, and we did that, and then you go back, let's say 2-3 months later, and you look at the entire body, no organs in these animals had an injury. You start to see green cells everywhere, in every organs of that animal. And if you look at different tissues, well, you have more in the liver than you have in the lung, more in the lung than you have in the heart, more in the heart than you have in the brain, but they are everywhere. So all across, what you can tell is that every organ is constantly in the process of tissue turnover. So they lose cells, and stem cells replace these cells. Same thing was seen in humans. People went to look at tissue biopsies coming from women who passed away years after receiving bone marrow transplant from a male donor. That means their bone marrow now contains the Y chromosome. So you can look throughout the body and see, how many cells do I have with the Y chromosome? The exact same phenomenon was seen. Out of all these studies, we can come out with this sort of general understanding. These numbers are not hard numbers, but you have a new liver every 2-3 years, a new pancreas 4-6 years, new heart 4-6 years, half of a new heart every 25 years. The whole body is constantly in a process of tissue turnover. Now, that's one part of the equation. The other part of the equation is to understand we're born with red marrow. As we age, red marrow converts to yellow marrow that does not make stem cells. By age 30, you have lost almost 90% of your red marrow. It's in your 30s that you discover you're no longer Superman or Superwoman, and now you start to get up in the morning realizing that there's some joints and some places, that are a little bit achy. That's because you have crossed that threshold where now you no longer have enough stem cells in circulation to offset cellular loss. From that point on, you start to accumulate a deficit. And that deficit, depending on your genetics, your life, past injuries, lifestyle, whatever, may show up in your heart, your liver, your lung, pancreas, whatever. But in 15 years down the road, it's one of those so-called age-related diseases. So when I published this concept, this understanding of the role of stem cells in disease formation, or the role of the lack of stem cells in disease formation, I published this 10 years ago, 2013. I thought there's one way to prove if this is real or not. Let's go and count the number of stem cells in the bloodstream of people who have developed those age-related diseases and compare that to healthy people. And now there's probably about 50 of those studies that have been done, and across the board: atherosclerosis, diabetes, liver failure, kidney failure, emphysema, heart disease, Parkinson, lupus, arthritis. The list is growing. Any one of those people who have developed those kind of diseases, they have on average less than half of the number of stem cells that we find in healthy people. Bottom line, how many stem cells you have in your blood today and tomorrow and the day after and every day for the rest of your life will determine how healthy you can remain, the health that you have today, how long you keep it as you age. That's the link between stem cells and health, wellness, and longevity.
Hey, super quick break from the podcast. Hopefully you're loving it. This is a way you can support the podcast and take advantage of a great offer. We're offering 50% off of one of our MCT bottles. MCT is an amazing oil. Add the ESS60 molecule to it. It's even more amazing. All you got to do is leave a review on iTunes, send us a notification that you've left that review. We'll get you a coupon code that'll get you 50% off of our MCT oil and free shipping. Back to the podcast. That makes me think, right, where we've got this transition, right, to the yellow marrow, right, and our stem cell count in our bloodstream has decreased. I've got some more questions about the components that increase the stem cells, but can we run out of stem cells? Like, is our certainly in the case of women and eggs, right, there's a fixed number of eggs when even in the embryonic state. Is there a fixed number of stem cells? Is there some limitation there?
There's not. There is a limit in the sense that your red marrow continues to shrink. So that bank of stem cells continues to shrink. But that rate of decline or conversion is not dependent upon how much you release. So it's just a normal shrinkage of that pool. How many people do you know who passed away because they ran out of stem cells?
Maybe a lot more than I realized.
We don't. There's a level of this that is true, meaning that as the number of stem cells decline, and there is this absolutely unavoidable decline in the number of circulating stem cells, there's a time when your tissue starts to not working well enough, and then you die of a deficiency of an organ, which at some point is linked to the deficiency in stem cells, but it's not linked to the fact that the red marrow that you have runs out. It's that your red marrow shrinks. The red marrow that you have right now, if you release stem cells from that red marrow, it will remain equal, like it maintains its homeostasis. If you release 10 million stem cells from your red marrow, you don't have 10 million stem cells fewer in your bone marrow. Yeah. They multiply and it maintains itself. So that is not a limitation.
And the way the human body tends to work, it wouldn't surprise me if you stimulate. I won't use the word overstimulate because that has different implications. But if you stimulate it, that it wouldn't surprise me if and you can correct me if I'm wrong, if you get some growth in that red marrow, because I think about how we build muscles. Like the way you build muscles is you break them down, and then, interesting to this conversation, stem cells go in there and repair them. So there is this kind of bounce back that comes off of it. That's pretty amazing. The other question I had, because I was looking, like I'm at your booth and I see you've got multiple stem cell stimulators in your product, and one of my thoughts is, well, if they're just have the same method of action, like the same thing is happening in order to release stem cells, then potentially you're just kind of piling stuff on there, and talk about that, because I know that's not the case, but talk about how you know that's not the case.
Well, and that's one of the beauty of this blend that I developed is that there are three main mechanisms of action in releasing stem cells, and the plants in there are doing one of those three different mechanisms of action. Some of the plants, we don't know what is the mechanism of action. But essentially, if I'm making this simple, because it is not simple, stem cells are maintained in the bone marrow by what is referred to as the SDF1 CXCR4 axis, which is a molecule called SDF1 that connects with a receptor CXCR4. When these connects, the stem cells releases molecules of adhesion and it clings to the environment. Anything that will disrupt that connection will release stem cells. Your natural signal when you have an injury is to release GCSF, that when it gets into the bone marrow, it triggers the release of an enzyme that digests SDF1. So when SDF1 is being digested and it's no longer there, you no longer have the connection, the adhesion molecules go away, the stem cell is released. The expression of the receptor happens with the activation of a protein called L-selectin. The blue-green algae contains a blocker of L-selectin. So if you don't express the receptor, you can have the SDF1 without the receptor, you lose that adhesion. That's one of the mechanisms of action. Other mechanism of action is to make your body react biologically as if you had an injury. So we can quantify in the blood an increase in GCSF. So it goes to your bone marrow and mimics as if you had an injury. So let's call them injury mimicking molecule. So some plants will do that. Panax will do something like this. Other compounds will stimulate in your blood. So you have in your bone marrow the SDF1 that is more concentrated than in the rest of your body to keep the stem cells in the bone marrow. You can increase the amount of SDF1 in your blood, and now it makes stem cells move toward the blood compartment. So other compounds, Panax works like this. So the plants all have different types of mechanism of action affecting this SDF1 CXCR4 axis at different level of its physiology. So the greatest likelihood here is that we have an amazing synergy, and that's what we see in people experiencing, this blend.
One of the things that I heard in one of your talks was a conversation about how different components that you knew were releasing stem cells were actually releasing them at different rates, right? So and for me, talk a little bit about that, because for me, I feel like you've come a long way from that simple understanding. Hey, if they're coming out at different rates, it's probably a different mechanism, right? I think that was maybe the initial piece, and then now you've actually got the research done to understand what are the, it's managing or interrupting SDF1 to CR4 connections, and you can do that on either end. Talk a little bit about that first kind of result where the rate was different.
Well, the first clue that it's a different mechanism of action is that you get a different dynamics of that curve. So the release with the blue-green algae was a release that peaks at 1 hour and then comes back down very quickly. Like at 3 hours after consumption, we're basically back to baseline level. So that's the blocker of L-selectin. You block L-selectin, the stem cells are released, and then that's it. There's like one wave. Other ingredients are going to build up those molecules that are mimicking an injury reaction. So as an injury, then it's more something that is on the long term. When you have an injury, the number of stem cells will slowly increase over a number of days. Here, the response is shorter, but we can see a release that goes and peaks at about like 3-4 hours and then comes back down to control level. And that was the clue to tell us that there are different mechanisms of action.
I loved, again, from the scientific perspective, those observations going, well, these must mean something different. When I was at the biohacking conference at your booth, I come to understand that one of the components in your product actually reduced the stem cells in the blood, so that there were less stem cells in the blood, but you still have it in your product. So talk to me about that.
Yeah, this is very interesting. We need to understand stem cell remain in the blood. Their residence time is roughly around an hour. So if you reduce the release of stem cells, it's not something that you will see in 20 or 30 minutes. So I'm saying this because as we're doing this work, so we're testing different things here. We're testing different plants. We come across like fucoidan, goji berry, great for longevity, medicinal mushroom. And as we start testing some of these plants, what we see is not an increase, is a very quick drop. 20 minutes after consumption, we see this drop in the number of stem cells in circulation. So the first reaction, and anybody who has been a scientist in a lab will tell you stories like these, you get something completely different from what you expect. So your first reaction is something is wrong. So we shut everything, we clean all the instruments, rerun the study, and we see the same result. Okay, the problem is elsewhere. So maybe our reagents, our antibodies are wrong and they've been corrupted. So you throw out your antibodies, you buy a new set of antibodies. It's expensive. Yeah. So, and then you rerun the test and you see the same thing. So now you start to step back and think maybe that's a real response. So as we dug more and more into this, what we realized is that famous CXCR4 receptor at the surface of a molecule that connects with SDF1 that keeps stem cells in the bone marrow, it's the same signal that is used by an injured tissue to attract stem cells in the tissue. So we realized that the density of these receptors increased dramatically after taking these polysaccharides, meaning the stem cells that is in the blood suddenly becomes much more responsive to the signal coming from tissues in need of repair. So they disappear from the blood as they migrate into tissues. So you see the decrease. So the moment that I discovered these plants and we could get a real sense here that it was a migration into tissues, then it's obviously common sense that if you put stem cells in circulation, but they do their job when they go into the tissue, that I'm going to blend them together so that I support them migrating into the tissues. So when I blend in these two together, then people trying the product would come back and would start to indeed say that they were getting better results. But it's hard to publish that kind of data because today in the scientific literature, migrating in a tissue, this process of migration is very poorly documented because there are no drugs in pharma that is used to stimulate that. So there's an absence of research. So we academically know the process, but there's very little research. So if I want to publish on it, then I need to go back more fundamental research to really document the whole process. Otherwise, it won't go through peer review. So I need to have funds I would love to do it. And I'm really looking forward to having the chance to do this because I am sure that it is one of those fundamental steps in healing. That is why many plants like medicinal mushroom, goji berry, a lot of these polysaccharides that have been documented to support health, historically, one of their main mechanism of action is just that. They support the migration of stem cells into tissues.
Out of marrow and into tissue, and that's again that kind of two part system. It seems like you could go back to your luminescent heart or luminescent muscle study and just look at it and say, well, how quickly did that happen? If we take one set of the rats and we pull some muscle tissue, how much of that has luminance versus one that doesn't have the component that's pushing those stem cells into the muscle tissue. That would be very cool. All right. So we've covered some great stuff. I want to ask, I like to get into some testimonials. I think, in the scientific community, and you and I are kind of first hardwired, don't listen to anecdotes, right? That's our first wiring. But the reality is anecdotes are an incredibly important part of the scientific process, right? Because most people think the scientific process starts at the hypothesis. But the hypothesis actually comes from the anecdotes, right? You don't start doing stem cell research until people are multiple phases, but you don't start doing research on the blue-green algae until people are reporting benefits. And you're like, okay, well, let's dig into this and try and figure that out. So so those people who have stem cells that are mobilized and potentially pushed into tissues, what are some of the kind of amazing, and this is going to be anecdotal, but what are some of the testimonials that you have around that process?
I mean, it's not just anecdotal. We have studies, ongoing studies in which we have cases. So it's being documented. But before I go to that, let me comment a little bit on what you just said, because I think this is a fundamental thing. Understand here, I am not saying that double-blind, placebo-controlled studies and the golden standard of science is not valid. It is extremely, not only valid, extremely important. But I do believe that it has taken a life of its own and it has become now almost ridiculous when you observe a fact of real life and you say that fact that I observe, I'm going to deny it because it did not happen in those 20 that were part of that study. To me, it's beyond ridiculous. The real answer is what was different in that one that was not in the other 20? That is my answer. So I use these to try to dig deeper and understand more than deny the reality, because right now this is really what is happening oftentimes in science. An anecdote is irrelevant if it's not part of a double-blind study, and I completely reject that concept. The anecdote, I totally accept it. I won't make any judgment on it. I'm not going to make hard conclusion on it, but I'm going to keep it in the back of my mind. And if I get 2, 3, 4, 5 of them, then I'm going to try to draw some sort of understanding, make a hypothesis, and I'm going to go and test the hypotheses. And I will even say that if my study to test the hypotheses shows that it's not working, I'm going to go back and say, what was different in these people that maybe I missed in my study before I really disregard the reality that I observed and was observable, or the real story of these people. You know, you get people sometimes having great results with a treatment or something, and then because it was not real in a study, you're basically telling these people, no, you still have your problem. The experience that you no longer have your problem is not real because in this study it was not proven. I find this so profoundly ludicrous. This being said, we are doing these studies. So we do have results. We have and by the way, in the so-called testimonial that we published, I have historically used testimonials with conditions that in science or in medical science are not known to reverse or to improve. Like you take, for example, somebody with a spinal cord injury, paralyzed, quadriplegic for 14, 15 years. Now we start to give that person this blend of plant that is known to stimulate stem cell release. If I see improvement in that person, you can tell me that it's not a double-blind study, but you'd be hard-pressed to not be forced to conclude that releasing your own stem cells obviously made a difference. So we have these cases published of quadriplegics getting mobility, stroke patients years after the stroke getting improvements, same thing with heart disease, same thing with many different kinds of conditions. Based on those observations in the past, we have started now studies. We're starting next month a study on Parkinson. We have an ongoing study on chronic stable congestive heart failure. So far in the study, it's ongoing. I'm writing right now a preliminary report on that study. And so far, all the participants in the study that started with chronic congestive heart failure 6 months after have normal cardiac function. So, and wow. I'm not saying that the plant or this blend of plant is good for congestive heart failure. All I'm saying is remember that little video that we talked about, how stem cells can repair the heart. That's all I'm talking about. Your stem cells can do it. All you need is have more in your blood circulation, so more are available to participate to that tissue repair, and that level of tissue repair becomes significant enough for you to see an improvement in your health. That's really just what the concept is.
Yeah, helping the body do what it does and taking that up a notch. That's pretty. This has been absolutely fascinating. I've got some wrap up questions. So, what are 2 to 3 protocols, habits, mindsets? Let's call them shortcuts because everybody likes shortcuts to longevity and a long health span that you would share, and maybe it's related to a blend of plant products, or maybe it's not.
Aside from stem cells, because I've been like so deep in stem cells right now, to me, any kind of health problems or lack or like not experiencing good longevity, it's because something is broken along the way. Something that is broken needs to repair. Your repair system are your stem cells. So at the core of absolutely anything that you want to do to improve your health, stem cells are the basics. So I've been so deep in this, I will say my first thing will be stem cell support. Let's put that aside. You asked the other 3. Probably my first one following this would be mindset. By mindset, I mean so many problem comes from and I know that a lot of people will say, don't tell me that my problem comes from my mind. It's a real problem. I'm not saying it's coming from your mind in the sense that you're making it up. What I mean is that it's the result of a thinking process that has an impact on the physiology that over years materialized into a problem. And the antidote for this to me is, we can call it so many different ways. Generally speaking, let me just say meditation. Just observe what your mind does, because 99% of what you think is not real. We're just making it up. The proof of this, something happens that is tragic. You take it, or somebody takes it really hard. The neighbor doesn't take it as hard. It's the proof that the event was not the cause of the hardship. It's how we take it. So just take a step back here. It's very easy to make life being like really sweet and soft, even in the midst of tragedy. So mindset, to me, is the most important. The second one, I would say, is probably exercise. The body has evolved over thousands of years with always having to move to be able to survive, gather your food. So it's almost like it evolved with movement. And here we are, sedentary like we have never been in history. Physical activity is tied to our physiology to a degree that we start to really understand it physiologically in terms of turning on longevity genes. And there's so many things, cognitive function, so many things that is dependent upon movement, blood circulation. So I would say move, physical activity, nothing intense, just good physical activity every day. And if there's a third one, I would say always stay on a 2/3 full stomach. Don't eat too much. Again,
like
our physiology evolved with always having to struggle for food. Our physiology is made to work well and when you're not full of food. And if there's one disease that we have in our Western world, is just we eat too much. It's the cause of so many problems. So I would say just try to half fast, and lean more under staying hungry than being full. So these are probably going to be like my three tops. And then there's tons of other things that we can do that are more targeted.
I love it. All right. So Christian, how can people contact you? What's the best way for them to connect with you?
Well, probably the best way, I'm on social media, Instagram, TikTok with Stem Cell Christian, because I think if I say Christian Drapeau, most people won't know how to pronounce or write my last name. So Stem Cell Christian, on our website, STEMREGEN.co, there's an email there. You can send queries. They will find their way to me or to my team. And yeah, that's probably the best way to reach me.
All right. And what in the near future, and you may have already touched on it, so if it's that, to bring it up again, what in the near future are you excited about?
When we release stem cells and they are in the blood circulation, how can we make them go more effectively to the brain or the heart or the liver or the pancreas, where we want where we want them to go? So start to stack now stem cell release technology with drivers of stem cells, red light, acupuncture, PMF, different technologies. So start to couple these in terms of research. That is sort of the next step, aside from the clinical trials that we're starting right now.
That's really exciting. All right. So you can find him. It's Stem Cell Christian. That's on social medias. Actually, I've been seeing your videos. I don't know if you ramped up recently or I just got connected with you, so I'm seeing them more often. That's very cool. And then STEMREGEN.co. That's STEMREGEN.co. Christian, thank you so much for joining us on this longevity summit.
My pleasure.
I hope you enjoyed this episode of the Live Beyond the Norms podcast. Make sure to subscribe and follow. Also, make sure not to be that guy, the guy sharing this podcast with people who are not interested in living beyond the norms. If you would like to learn more about the ESS60 molecule, the molecule responsible for the single longest longevity experimental result on mammals in history, a full 90% extension of life, visit myvitalc.com. That's myvitalc as in carbon.com.